Hello, I'm Lilah Mehta, and I'm presenting my project on cell type specific ERK hyperactivation and how it promotes inflammatory remodeling that may worsen neuroinflammation in Alzheimer's. Alzheimer's disease, or AD, affects over 55 million patients worldwide and is understood not only as a neuronal disorder, but as one driven by dysregulated immune signaling. One signaling pathway that plays a central role in this process is the extracellular signal regulated kinase, or ERK pathway, which is part of the MAPK family of proteins. ERK signaling follows a RAS to RAF to MEK to ERK cascade and regulates immune activation. In AD, elevated ERK has been linked to disease-associated microglia, or DAMs. And in late AD stages, DAMs shift from protective and anti-inflammatory to pathological, and ERK drives DAM gene expression and neuroinflammation. CX3CR1, a chemokine receptor, is not just expressed on microglia but also on peripheral myeloid cells. Because these immune cells share developmental origins, I wanted to know whether sustained ERK hyperactivation in CX3CR1 positive immune cells could reshape peripheral inflammation in ways that mirror or contribute to neuroinflammatory mechanisms observed in AD. To test this, my lab generated a BRAF B600E mouse model that causes chronic ERK hyperactivation specifically in CX3CR1 cells. I analyzed spleen tissue from 19 spleens using mass spectrometry to compare protein expression at baseline and under inflammatory stress induced by LPS. Despite the complexity of thousands of differentially expressed proteins, the overall pattern was clear. ERK hyperactivation primes the immune system toward a pro-inflammatory state. Using Ingenuity Pathway, I also generated a causal network showing inhibition of homeostatic immune pathways and strong activation of ERK-related enzymes, reinforcing the idea that ERK hyperactivation removes normal immune constraints. I also confirmed increased ISG15 by Western blot. My results are significant because peripheral immune activation can drive neuroinflammation via cytokine and complement signaling across the blood-brain barrier. ERK may be a shared inflammatory regulator across peripheral immune cells and the CNS, and many of the inflammatory pathways upregulated in my model overlap with those defining DAMs in AD. In ongoing work, ERK suppression using a PD-MEK inhibitor also reduced ERK activation in the 5x FED Alzheimer's mouse brain, and this supports the feasibility of targeting ERK signaling as a potential therapeutic target. In sum, this study identifies ERK as a central regulator of immune balance and shows that elevated ERK drives systemic inflammation that may mirror or contribute to neuroinflammation in AD. Thank you for listening.